Thanks.
All right, thank you. Yeah, so thank you everybody for attending this webinar on the distribution, status assessment, and population ecology of Cagle's map turtle. So this research was funded as a state wildlife grant, Section 6, so with financial backing from U.S. Fish and Wildlife Service. And yeah, so I'm going to basically discuss a little bit about our 4 years of research on Cagle's map turtles, which I think has been really exciting and hopefully very interesting for you all as well. And so before I really get into this, though, I think I really need to thank several people who've really made this research possible. The obviously, given that it's such a lengthy study over 4 years, that we've had a lot of involvement from other people. In particular, this top row of students, Brooke, Isabella, Sarah, and Lexi, have all been involved very extensively in this research in the field for at least 2 seasons, for at least 2 years. Brooke, in particular, has also been involved in helping us make some maps for our report and projects, so thank you. The second row, we have Clint and some other people who have helped for at least a season in the field, and I would like to thank Peter Lindemann, who is easily the world's leading expert on map turtles, who very graciously has come on board to assist in this research and advising in all things map turtle, and his knowledge is just impressive, and we're very lucky to have been able to tap into that. And I'd also like to thank Nate Bendick for not only helping me in the field with some recon early on this project, but also for um, basically sharing his ideas and thoughts on hierarchical modeling, which is something that we'll talk about today. And of course, there's a lot of other people listed here below that have helped in one way or another. So thank you all. And yeah, with that, let me discuss a little bit, give you a little background on Cagle's map turtle. All right, so Cagle's map turtle is Graptemys caglei, is one of about 14 species of map turtles that are collectively one of the most species-rich groups of emydid turtles and found throughout eastern North America. Some of these species are quite widespread, but for the most part, many Graptemys are drainage endemics that have highly restricted distributions. And one example of this, of course, is Cagle's map turtle, which is endemic to the Guadalupe, San Marcos, and Blanco, basically rivers here, as you can see on this map, kind of orienting you to where our study area is. These turtles were described fairly recently. In fact, we're at the 50th anniversary right now. And so I think it's kind of a very special time. 50 years into only 50 years into knowing that this species exists, an interesting time to kind of look back at the history of research on this turtle and kind of give an update on its status, its distribution, and abundance. You can see here, like I said, if it's the southwesternmost species, and it's found in over a distance, river lengthwise, of only about 600 miles total. So you can see we can kind of consider it to be a micro-endemic. These are some pictures of Cagle's map turtle, showing you kind of their distinctive markings. For example, you can see that from these pictures that Cagle's map turtle has a gray or kind of black head with white or kind of pale yellow markings, stripes, kind of ocelli, a little kind of interesting head pattern. You can see on the picture on the right, you could see the back of the head, the dorsal view of the head here, where you see a series of kind of post-orbital bars or chevrons, which give this turtle what we call its common name for us, which we call the Whataburger turtle. It kind of looks like a Whataburger logo. Also, you see on this other guy, you see kind of this transverse white bar below the chin, this mandibular bar. This is a unique trait, a unique characteristic for Cagle's map turtle that kind of differentiates it from other turtles, other map turtles occurring in Texas, which generally have mandibular spots or ocelli. So also you can kind of see the distinctive notches or the kind of vertebral spines or knobs that you see along the carapace here that are also characteristic of map turtles. And you can kind of see the slightly bulgy costal shell elements that are also kind of characteristic of this group of turtles, which are highly riverine, highly aquatic. They very rarely move on land, disperse over land. My controls aren't working here very well. Sorry. Um, if you learn about map turtles, probably the most interesting first kind of thing that you would notice about them that's really kind of cool is the extreme sexual size dimorphism. It is actually in probably one of the most extremes in terms of vertebrate diversity. For example, you might think of something iconic in vertebrates. I'm thinking elephant seals, where males can attain weights of about 3 times females. Well, map turtles have them beat. It turns out that females outweigh males by about an order of magnitude. All right, so male map turtles are often about 100 grams, whereas females can get easily over 1,000 grams. This picture on the right, this was really fascinating to me. I saw these map turtles kind of interacting. You can see the male facing the female, kind of getting in her face, and he kept kind of fluttering his forearms a bit, kind of flashing her, which basically this is an initial step in courtship where the male is kind of convincing the female based on his markings that he belongs to the same species. So this is likely establishing visual conspecific recognition as an initial step in courtship. Here are some more obligatory pictures of male and female Cagle's map turtles. These are some individuals we caught near Cuero. You can once again kind of see the impressive difference in size. Here's another picture. I like this one. It's kind of a stately portrait of, you've got the male on the left and you've got the female on the right. Once again, you can see the notable difference in size. In many cases, when people first see map turtles, they think the males are juveniles or babies, which is kind of fun, but they're not. This is a full-grown male. And another feature of map turtle dimorphism is the tail length. For example, here, this is a great picture because on the left you see a juvenile female that's about the same size as the map turtle on the right, which is a male. And notice the size of the male's tail versus the female's tail. So she has a little nubbin of a tail where he has a quite enlarged tail, even though you can see for this particular picture, these individuals are roughly the same size, this juvenile female and adult male. Also, everybody wants to see the babies, of course. Here are some photos of hatchling Cagle's map turtles. They start out about the size of a quarter. And at this stage, it's not particularly easy. In fact, pretty much impossible unless you probably were to do a dissection, but to distinguish the males from the females. But you can see that, as you probably expected, with how gorgeous the adult turtles are, the juveniles and are quite adorable as well. A couple other things about map turtles. They're surprisingly cryptic and they're extremely social. They're gregarious animals and they're exceptional climbers. So they can climb essentially vertical tree limbs. I've seen them as high as 20 feet up above the water. And if you're a surveyor looking for map turtles and are not prepared for this, they can be very difficult to spot. They're surprisingly hard to see, despite the fact that they're basically these little discs that look like they're plastered to a rock or a log at any point in time. They're obligatory baskers that will essentially use any substrate, rock or log, for the most part, as long as it extends away from the shoreline. And in this respect, they differ from a lot of the sympatric turtles that are in the Guadalupe system, which will often bask on the shorelines. These turtles are exceptionally wary and they tend to not bask on the shorelines. I'm thinking in about 3,000 observations of Cagle's map turtles, I could count on probably both my hands the number of times I've actually seen them basking on shores or on trees that are actually growing on the shore. Usually, if you see something if it's basking on a substrate that's connected to the shore, that can sometimes happen as long as it's mostly surrounded by water. Here on the below on the right, you can see, for example, what look like these smooth little paving stones. These are pictures of, for example, spiny softshell turtles. We see softshell turtles, cooters, all of these kind of other turtles have no qualms about basking on the shoreline. Not so for map turtles. Before we get too far along in this, I just want to kind of point out some other players in the story. We have various turtles that occur in the Guadalupe River drainage, in addition to Cagle's map turtle. For example, we have pond sliders and Texas river cooters. Texas river cooters, you could see in the bottom left here in this picture, along with a Cagle's map turtle. These are the most, clearly the most dominant turtle in this river system, the river cooter. But we do have sliders, we do have spiny soft shells, we have stink pots. I've occasionally, only on a couple occasions have I seen common snapping turtles, but they're in the Guadalupe River as well. In fact, this is kind of a funny little anecdote. This picture of the river you see here, this is actually Lake Gonzales right after the dam failed. So this is about 3 days after the failure of the dam. So we went out here and did some surveys for turtles, and apparently the draining of this reservoir completely discombobulated all these stink pots, and we were able to see them kind of wandering around in what appeared to be kind of a confused state. Um, anyway, yeah, so these are some other turtles that I'm going to discuss as we kind of move through this webinar. So pretty much most of the limited information that we have on Cagle's map turtle and knowledge on the biology of the species comes from a large body of gray literature and theses that were directed by Flavius Killebrew, all right, in his lab out of West Texas A&M University. These are probably the most extensive studies of map turtles to date. They studied the species for about, sorry, not map turtles, but excuse me, Cagle's map turtles. And they studied the species for about 15 years and produced a lot of reports related to their findings. And based on their studies, some of the major inferences, or I want to kind of say dogma, on the biology of Graptemys caglei are pretty much at the time that they completed their studies, we knew a little bit about their populations and distribution, where they determined that there was a core population of Cagle's map turtles basically occurring from Seguin to Cuero, and that the population declined toward Victoria. Also, upstream of Seguin, they considered Graptemys caglei to exist in extremely small and fragmented populations, basically all the way up to Kerrville, and also have some very small populations in the lower San Marcos River. They also did some surveys in the San Antonio River and considered Cagle's map turtle to be extirpated from the San Antonio River. And I'll discuss this in a minute here. But probably one of the most important things is that they actually came up with an estimate of global population size for Cagle's map turtle. And they determined that within the entire river system, there are about 13,000 map turtles at the time. And this was based on doing single pass surveys along the river, but using kind of applying a correction factor that they termed an index of conspicuousness. Essentially what happened, how they obtained this index of conspicuousness, which is essentially a type of detection probability, is they had a study area of about, of a short stretch of river, let's say about 20 kilometers, and they did a long-term recapture study on this population and determined, let's just say, for example, this isn't the exact numbers, but this isn't the numbers at all, but let's just say they determined there were 1,000 turtles there. Then they would float that same stretch of river and let's say they counted 100 turtles. So they would say that their index of conspicuousness is 0.1 or 10%. They saw 10% of the turtles there. So they use that kind of index as a metric to basically apply to single pass surveys across the entire river drainage to come up with this global estimate of abundance. And the thing that's important here is that their detection probability was extremely low. They determined that they only saw about 3% of the turtles that were actually on that particular stretch of river. They also did a lot of work on the diet of Cagle's map turtles, all right, and noticed, and which was, I think, their best work, really. They did a lot of diet studies and determined that the males feed primarily on aquatic insects, the nymphs of trichopterans and odonates. Ephemeropterans as well made up a large proportion of the diet, and that the females fed primarily on mollusks, and in particular the invasive Asiatic clams. So made up about 90% of the diet of females by weight. And they also did some work on habitat associations with Cagle's map turtles, where they kind of I think this work is a little bit problematic, if you read it closely. But essentially, it's kind of persisted as, kind of the biology of Cagle's map turtle for the last 30 years or so. But they basically determined that Cagle's map turtle required natural riverine habitats that included riffles, runs, and pools, that these turtles were absent from large impoundments. There's a chain of impoundments on the Guadalupe River extending from New Braunfels to Seguin, and they were unable to detect any map turtles in those impoundments, or neither for Canyon Lake as well. So they kind of basically decided that Cagle's map turtle required at least some moderate water velocity. They required some level of water flow. And they also assumed that, or made the kind of, determined that Cagle's map turtles were associated with black willow. So willow was an important component of the riparian zone where these turtles existed and reached high abundance. And they attributed this to their observation that the willow roots supported large quantities of trichopteran larvae. So another kind of a little quirk of history relating to the distribution of Cagle's map turtles comes from the earliest publication, the description of the species, where Haynes and McCown described the Cagle's map turtle and presented a figure that you see here of the various drainage basins that are occupied by different species of map turtles. And even though in the figure they emphasized that they were demarcating drainages and not the exact distributions of these turtles per se, right? This kind of ended up creating, along with some contradictory passages in their article, kind of created a 50-year confusion as to whether or not Cagle's map turtles actually inhabit the San Antonio River drainage. And the San Antonio River is the drainage to the south of the Guadalupe, and it's actually connected with the Guadalupe River drainage. So it forks with the Guadalupe about 10 miles upstream from the mouth of the Guadalupe River at San Antonio Bay. So these are, I kind of excised here two important passages that kind of unfortunately resulted in this confusion. All right, so it says if you look here at the kind of the middle passage here, it says Graptemys caglei is abundant at the type locality and in other sections of the Guadalupe River, the San Antonio River, and several smaller tributaries. And then in a later section of the paper, they said Graptemys caglei has been infrequently collected in the San Marcos River. Intensive collecting of both the San Antonio and Medina rivers has yielded only a few site records. So they made these two kind of contradictory statements. On the one hand, said it was common in sections of the San Antonio River, and then only based on a few site records from the San Antonio River. And Haynes later kind of corrected this mistake and kind of confined the occurrence of Cagle's map turtle to a single site record at the Mission Street Bridge in the south side of San Antonio. And however, the damage had already kind of been done in the sense that I think these statements kind of set off 50 years of uncertainty regarding whether or not Graptemys caglei does occur in the San Antonio River. A lot of popular sources, I'm looking here at the left with the Field Guide of Texas Turtles, and of course, Peter's book on map turtles. These books, I think, quite accurately, kind of describe the situation and emphasize that there was actually really no evidence of the species ever really occurring in the SAM drainage, SAM meaning San Antonio, Medina. But there are also other popular sources that you see here on the right that kind of advocated a view of Graptemys caglei having once occurred in the San Antonio drainage and now being extirpated, right? And this also view was propagated also by Killebrew's team as they studied map turtles in the 80s and 90s. So, and in some instances, the extinction of caglei, the supposed extinction of caglei from the San Antonio River was even speculated to have to do with pollution of the San Antonio River. So it's kind of interesting. The speculation kind of got out of control, which I think, kind of happens when you have very little actual information to work with. And so this leads us a little bit to the status and conservation history of Cagle's map turtle. So based on all of this work, there have been a lot of historical concerns regarding the status of the species, for example, because it is in essence a micro-endemic restricted to the Guadalupe River drainage, and at the time was believed to even be highly restricted within the river system itself. Also, there were concerns about the fragmentation of habitat, in particular water diversion and water quality, particularly because of the conception that these map turtles did not do well in reservoirs. And also at the time, there were a lot of plans for the construction of additional reservoirs in the Guadalupe River system. Human depredation, as well as just general presumed declines in abundance. So from these concerns, Cagle's map turtle was considered a candidate for federal listing between 1982 and 2006. However, because the state of Texas opted to list Cagle's map turtle as state threatened in 2000, thereby offering the turtle protection, it was no longer really needed to be considered at the federal level. And even recently, the IUCN Red List status of Cagle's map turtle is endangered or highly endangered. And a direct quote from that assessment is that the range has been reduced by half to two-thirds since 1974. And that statement directly comes from the belief that Cagle's map turtle was, I guess, completely distributed throughout the San Antonio River and then has since become extirpated from that system. So, assuming that that's incorrect, then, there's absolutely no loss of distribution since that time. However, it's played a heavy role in kind of considerations of the conservation and status of the species since then. So this brings us to what we did. So we did a 4-year survey-based study of abundance and distribution of Cagle's map turtle. And by abundance, we're referring to the number of organisms, right, in a specific location. And distribution is it kind of a reduced information version of abundance where we're looking at where the species occurs, right? So this deals with where the species occurs. And our study is based entirely on a new analytical framework that was developed over the past 20 years, which incorporates what is called a metapopulation sampling design. So this type of sampling is really straightforward, at least conceptually, although it can be kind of difficult to execute. But all it really means is that we are replicating surveys, both temporally and spatially. All right, so we're going to get replicates over space, preferably over the entire distribution of a species or what would be our study area. And we want to repeat those surveys on several occasions. All right, so the reason being is that by basically doing this type of metapopulation sampling design, it allows us to analyze the data using a hierarchical modeling framework in which we can basically use the variance in counts, for example, between surveys or detections to basically build up a detection probability estimate without actually having to mark or otherwise identify individuals. So in essence, this type of detection probability is conditioned on count data. That's why this would be considered a hierarchical model, because it basically is a generalized linear mix model where the sites basically form a natural kind of random effect, but you also have submodel for detection in addition to counts. So we generally, in popular literature, we call these N mixture models, all right? And these are essentially, like I said, just generalized linear mix models that are modeling count data with an additional submodel for basically for detection probability, right? So these are the main parameters of the models that we are looking to estimate. So P is the detection probability. This is usually modeled as a binomial distribution. So if you were to imagine, for example, if we went to a pond and we knew there were 10 turtles there, and we happened to see 3 of those, that means we had 3 successes and 7 failures, right? So for this type of data or conceptualization of data is readily accommodated by the binomial distribution. So this is kind of a canonical distribution for modeling detection. And then we also are interested in lambda, which is the mean counts of a survey, right? So we usually model counts based on a Poisson or negative binomial distribution. And yeah, so together, basically, we can perform these end mixture models to obtain some estimates of abundance without requirement that the individuals be marked. And in addition, we can also, like any other sort of modeling approach, we can incorporate covariates in each of these submodels, so that both the detection and the abundance submodel, so that we can evaluate, for example, habitat characteristics that might explain heterogeneity in our count data, or also heterogeneity in our detection data. So that makes them extremely, extremely useful. And so we used Ned mixture models to evaluate abundance, looking at sampling, a metapopulation sampling design over the known distribution of the species, which is the GSM, or what I'm calling the Guadalupe-San Marcos drainage is GSM. So when we move over to distribution, we're going to expand our study area to look at the entire drainage, including the Guadalupe-San Marcos, as well as the San Antonio-Medina drainage. So essentially the potential distribution. And what I'm hoping to convince you here is that this type of analysis is exactly the same as an end mixture analysis, just incorporating different distributions and different types of input data. So once again, this is a metapopulation sampling design, where for our study, we replicated point surveys from bridges. And our n equals 5 here means we did 5 actual surveys of each site from a bridge, which is kind of different from what we did for our abundance models, where we replicated 3 transect surveys from kayak. So we were actually on the river for these transect surveys. We did 3 per survey for the abundance models. For the bridges, we did 5. So similarly to the same to the abundance models, this allows a hierarchical modeling framework in which our detection probability can be conditioned on presence-absence data. So these are typically called occupancy models. And once again, we're going to model P, which is the detection probability, also on the binomial distribution, and psi, which is the mean occupancy, or the proportion of sites occupied, which is also modeled with a binomial distribution. Once again, this does not require the animals to be marked or otherwise identified, and likewise can incorporate covariates in the submodels. So there are some assumptions of performing these models on abundance and occupancy. One of which is has to be population closure, all right, which means that there's no addition or removal of individuals from the area during the time of the surveys, either by migration or by predation or whatnot. So in order to kind of ameliorate this assumption, we attempted to survey all of our transects on consecutive days. So this kind of closed that window. It helped make sure that we can adhere to this closure assumption. We did not do this for the occupancy models because the closure assumption is less stringent at that point, because with occupancy models, the detection probability is simply the probability that you will detect the species, and hence the closure has to do with whether or not the species is still present in the site. So this differs a little bit from the end mixture models, where the detection probability is actually interpreted as the probability of encountering the same individual of a species. So this closure assumption matters a little bit more. Another important assumption is that you make no false positives. So we tried to ameliorate this by requiring voucher photographs for every transect and every site. And we also, if there was a question on what a turtle is, what it was, we don't mark unknown turtles or try to guess on turtles. We simply did not count or enumerate unidentified turtles and assumed that basically these turtles would be kind of absorbed in the detection submodel to basically kind of inform our detection. Also, independence of detection of individuals is extremely important, and this is probably where we violated the assumption most heinously because, as it turns out, these turtles are extremely gregarious, extremely social, and we see them mostly in groups. All right, so there's clearly violates this. Um, on the other hand, you can kind of account for this somewhat by fitting our distribution with a negative binomial distribution, which essentially accounts for over-dispersed or clumped data. And the other kind of major assumption of these models is constant detection probability, or at least that your detection probability is modeled adequately. And this here, if you're a purist in terms of modeling, this is the hardest assumption to really overcome, partly because detection probability itself is a function of abundance, right? So you're trying to determine a parameter, you're trying to estimate an abundance parameter based on detection, but at the same time, that parameter is going to vary as abundance increases. Obviously, detection also will increase. So anyway, this is just kind of a little bit of an overview on the assumptions of these models before I show you kind of the results. I'm not going to get into a lot for the sake of time, our protocols for surveys themselves, but I'm going to tell you that we were highly dependent on optics. So we relied very heavily on cameras to do our surveys, to make sure we had accurate counts. Can't tell you how many times I've pored over photographs after particular surveys to, make sure that our counts are as accurate as possible. Because what happens is sometimes you'll see aggregations of turtles like you see here. And you're looking in binoculars, which aren't very stable. You're trying to make some counts. So if you can able to snap off a quick shot, then you can at least go back over and kind of verify those counts. Like, for example, if we were to play a little game here, can you tell me how many Cagle's map turtles are on this log? I'm going to count for you. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 Cagle's map turtles. Obviously, counting this many would be very difficult if if we were just there in situ. So having these photographs kind of helps things a lot. So with that, let me talk a little bit about the abundance models first, and I'll tell you about the results of these models. And then after that, we will move on and talk about the occupancy models. So for our abundance models, we also performed parallel analyses on Texas river cooter from the Guadalupe-San Marcos drainage. And we did this for a couple of reasons. First of all, we thought that it would add comparative value to our study, but also for contextual reasons, because it would allow us to help us to piece together and infer population changes that occurred over the past 25 years since the Killebrew work, because these researchers had also considered abundance data on river cooters in their analyses as well. So this kind of can help give us a little bit of context for how populations had changed over 25 years since the Killebrew study. So just so you can kind of have an idea of the various types of covariates that we included in our models, we had, for detection covariates, we primarily used climatological data that we measured on site while we were taking our surveys. But we also included some time data, for example, time since sunrise in minutes that we obtained from the NOAA solar calculator, solar clock calculator, and also time until sunset, ordinal date, cloud cover, and various other kind of measures, humidity, temperature, station pressure. And we also looked at some various site covariates. So the site covariates relate more to abundance, whereas the detection covariates, of course, as the name implies, relate to whether or not we're going to detect a turtle at a site. So these included river sinuosity, the channel width, which is basically a kind of a crude surrogate of water volume in a sense. And we also did a lot of categorical variables. So we looked at dominant woody vegetation as 3 levels, whether it was willow dominant, cypress dominant, or mixed hardwood. We looked at the riverine habitat, whether it was river runs pool, so kind of unmodified river, whether we were in an impoundment, or whether the transect included both impoundment stretches as well as riverine stretches. We also looked at terrestrial soil in 4 categories, so whether it was clay, loam, rocky, loamy bottomland or sandy mix of soils. We looked at riverbank slopes. We looked at river substrates, also a category with 4 levels: bedrock, large and small gravel, sandy or silty to sandy. And to kind of get a grasp on any sort of potential interspecific competition effects, we also evaluated mean counts of other emydid turtles from our surveys and looked at that, included those in models too. So to give you kind of an idea of, for example, dominant woody vegetation, these are the 3 kind of images of the 3 categories. We have on the left here, you have the mixed hardwoods on the Guadalupe River near Gonzales. Typically, when we had mixed hardwood, transects that were mixed hardwood, they were dominated mostly by pecan or sycamore or green ash or box elder. And then we looked also at whether the sites were dominated by willow, as you can see here in the middle on the San Antonio River, and cypress dominant, as you can see on the right. So what kind of modeling procedures did we do here? For our analysis for end mixture models, I'm just going to run through this pretty quickly just so you kind of have an idea of how we approached the modeling, but it's fairly complex. But for now, what we did we initially did preliminary models in a maximum likelihood framework, all right, using the program or package, our package Unmarked, which is designed specifically to deal with these types of hierarchical models where we're not actually marking individuals. Because sample size limits our ability to parameterize our model, we had to kind of limit complexity up to 15 total parameters because we noticed that this was kind of the level at which the model would break, so to speak. And so we limited complexity to about 15 total parameters, which seems like a lot, but you got to remember that we also, for a lot of our variables that were continuous, we looked at, for example, quadratic or cubic, in addition to kind of linear functions, which all cost parameters. We do not include, we standardized continuous covariates and scaled them. We did not include strongly correlated variables in the same models. So we kind of had those major kind of constraints on modeling. We also designated, to kind of account for spatial structuring in our data, we encoded 2 different geography blocks. So one basically included the transects as broken down by EPA level 3 ecoregions, right? So that includes Edwards Plateau, Texas Blackland Prairie, East Central Texas Plains, and the Western Gulf Coastal Plain. And the other block that we called the physiography and hydrology block basically was based on the forking patterns of the river, the hydrology, how much water was in certain stretches of the river, and those kind of major kind of physiographical features. So we also included transect length as an offset, and we did this because our transects for our surveys varied in length from 1 to 12 kilometers, unfortunately. Which required that we kind of dealt with that in one way or another. Another option is to model the transect length directly as a covariate, but we use an offset, and what this does is it shifts our interpretation of abundance to density, all right, to number of turtles per river kilometer. And we also, of course, evaluated the choice of mixture distribution. And you can tell from these histograms on the screen here, this is our raw count data. On the left is for Cagle's map turtle, on the right is for Texas river cooter. You can see kind of highly dispersed, and at least for Cagle's map turtle, that data is also 0 inflated. For Texas river cooter, there's a lot of low counts, but it's not 0 inflated. It's just highly dispersed. So anyway, we presume that this would be negative binomial distribution would be the most appropriate mixture, and it was. And of course, we compared all model performances using AIC to basically figure out which of our series of candidate models contain the most information, or in other words, the best fit given our data. And then what happened is we ran into a problem. All right, the problem was that it turns out that unmarked, as it implements the negative binomial distribution using maximum likelihood, didn't really work so well, because what happened is we had unidentifiable parameter estimates. If we increased basically we increase this parameter, which is the summation limit for integrating over the latent abundant state, which is called K. This is kind of a parameter you have to feed into unmarked. If we anytime we increase this parameter, this K, what would happen is we would increase our abundance estimates. So it was unstable. What this meant is that we kind of moved our final analyses into a Bayesian framework using another R package called SP Abundance, which basically um, which allows us to kind of circumvent this problem. And here we use three complementary models for each species. Each incorporated sunrise and cloud cover in the detection covariates, because for both Cagle's map turtles and river cooters, sunrise and cloud cover were the most important and most informative detection parameters or detection covariables. And we included transect length as an offset. And we did three sets of models. One where we used our geography blocks as a fixed effect for abundance, where we looked at our top abundance covariates as fixed effects for each species, and the top abundance covariates plus the inclusion of geography as a random effect. And we checked all of these models to make sure that the MCMC, the Markov chains converged and mixed well. We ranked our models and assessed model fits. So moving on here, if I can do this. I'm running into a problem. My slides aren't advancing. Here we go. All right, so here, just so you can look at our kind of physiography and hydrology blocks for the river, you can see we separated the river into the Guadalupe River into three sections: the headwaters, which included kind of perennial water, because this is where they're kind of fed by springs. We included the middle Guadalupe, which both of these were on the Edwards Plateau. The middle Guadalupe, this part of the river experiences a lot of drought and low flows. And then we had the lower Guadalupe, which is basically the entire river below the Balcones Escarpment, which is kind of marked here with a dash. Then, of course, we had the Blanco River and the San Marcos River, too, in our geography blocks. So you can see anything above on the Edwards Plateau, the river is quite distinctive. It looks very different. Flows are limited. The water is clear. A lot of bedrock, limestone cliffs, and escarpments. And then below, on the actual coastal plains, you have a much different kind of characteristic, different type of river, right? You have a kind of a slow, sluggish, meandering but wide and high water volume kind of river. So during our surveys, during our study, we experienced major drought conditions. So we saw this kind of created hindrances for our surveying and also our study design. This had the lowest discharge rates of water in the Guadalupe since the 1950s, which was a major drought of the last century. We've also seen during our study the lowest levels at Canyon Lake since Canyon Lake was impounded in 1964. So this kind of created a lot of issues with our surveying, but it also created some opportunities because it provided us a unique perspective on kind of the ecology of drought in extreme conditions. We would see, for example, large mass graves of mussel beds where water level had dropped to the point that they were exposed and died. We saw this one mussel here, see it making a little circle. That's part of an SOS, begging for help to get it into better conditions. But anyway, we had a lot of drought conditions, which I think kind of I'm gonna circle back to at the end here. But let me talk a little bit about the results of our abundance analyses. So so you can understand kind of the scope of this study, we did 187 surveys across 74 transects. The mean transect length was about 5 kilometers, and it amounted to a total of just under 400 kilometers of unique river coverage, and ultimately just under 1,000 total kilometers surveyed. During our surveys, we observed 12,000 plus turtle observations. About 20% of these, or 2,400, were Cagle's map turtles. About 8,000 were Texas river cooters, and then the remaining about 1,420 turtles were combined pond sliders, spiny softshells, stink pots, and also Texas map turtles, which we didn't know before this study, but had apparently invaded into the upper Guadalupe River system, and where we have found them in our surveys. So these red lines demarcate kind of our survey locations or transects. You can see that we've did our best to, given the constraints of access and private land in Texas, we did our best to try to spread our transects across the entire system as best as possible. What about our best fit models? For Cagle's map turtle, it was the detection covariates were time since sunrise and cloud cover. For abundance, it was the dominant woody vegetation and the terrestrial soil categories. And we obviously included a transect length offset, and also our best model included a geography block. For Texas river cooter, the detection variables that were in the top model included the time since sunrise, the cloud cover, and abundance was determined also by dominant woody vegetation, but rather also river substrate, and no geography blocks. So basically, for Texas cooter, since the geography blocks weren't included in the best fit models, we determined that geography was a relatively poor predictor of abundance. So here, these plots here show the detection probability, all right, for Cagle's map turtle on the left and for Texas river cooters on the right. So this is pretty cool because our detection submodels indicated daily peaks in basking probability, as well as increased detection under intermediate cloud conditions. So this suggests that kind of conserved diel thermal regulatory behaviors are found in both of these turtles, right? So both of them are basically cueing their activity on both time cues, I guess where the sun would be more or less, and also with cloud cover. So as you can see here, activity for Cagle's map turtle peaked at about 400 minutes after sunrise, whereas for Texas river cooter, this was about 50 minutes or about an hour earlier, which holds with our data. Also, we noticed that there were kind of peaks in cloud cover activity at intermediate cloud cover, which was also important in explaining detection. But what's really interesting to me is if you can see on the left, you can see the confidence intervals around, or the credible intervals, I should say, around the detection probability for Cagle's map turtle are much broader than for Texas river cooters. So this indicates that river cooters are more precise thermal regulators. They're more constrained to basking at very specific times and under very specific conditions. And this makes sense because Texas river cooters are larger and they're also herbivorous, so they have some extra kind of digestive energy requirements. The kind of cool thing about this to me is I think that if you see the immediate, especially when you're looking at kind of the immediate, where am I going here? If you look at the, excuse me, right? If you look at kind of the intermediate peak in detection on cloud cover, kind of indicates to me that this is kind of a Goldilocks zone in terms of detection, because at low cloud cover, the required length of time for basking is really short, and these are, the turtle can jump in the water at a far distance if it sees you from afar. Whereas if it's extremely cloudy, of course, there's very minimal benefit to basking at all. So at this intermediate level, this Goldilocks zone, it seems that if the turtle will be kind of hesitant to jump off its basking substrate unless you get a little bit closer, which helps our detection. So it's you can kind of see this kind of trade-off between, meeting these particular thermal regulatory requirements at the same time avoiding predation. But I think this also contributes to the general perception of map turtles as as warier turtles than river cooters because, they're not as constrained to thermal regulate, at a particular time and for a particular length of time or under particular conditions. So they have a lot more flexibility there. So they can kind of afford to be um, um, more, I guess, more wary, more discriminate about jumping in the water and leaving their basking perch at the slightest threat. So I thought that was very interesting. Also, kind of led me to think a lot about the fact that we see turtle stacking all the time. So Cagle's map turtles, particularly these males, will tend to stack on top of other turtles, even when they have plenty of other available substrate to use. So it's tough to understand. I don't know exactly what's causing this, whether it's some kind of micro conditions, whether it's some type of social interaction, or another option is that they could be lookouts potentially, given that they are wary and that they could be kind of looking out. And if they jump in the water, this could be an indicator to these other sliders and cooters that there's a threat nearby. And yeah, so there's could basically be some kind of interesting behavior that could be studied further. So I'm going to very briefly explain for our best fit models what were the important variables and kind of what it means to explaining heterogeneity and abundance. Basically, our best fitting models indicated that map turtles showed highest densities in basically in mixed hardwood, all right, habitats, or sorry, vegetation types. And also, though, this overlaps strongly with willow habitats as well. But you could see that it's pretty much better interpreted as a avoidance of cypress habitat, as cypress vegetation. And they also showed a positive association with abundance in loamy bottomland and sandy substrates. So together, these habitat effects suggest to us that what's kind of influencing abundance of map turtles is specifically the availability of nesting substrates. All right, so they prefer to nest in loam or sand, and the fact is sand didn't really show up as a very strong predictor because we have very low power with sand. We didn't have a lot of replication on sand because the Guadalupe is more of a gravelly river. It's not really a sandy river. So I imagine if there was more sand, we would see this effect much more strongly. All right. And as far as why are they avoiding cypress, we think it could probably be also some indirect and subtle effects on nesting ability or nesting as success as well, given that cypress trees tend to inhabit rockier soils. So there could be some confounding of soil type with vegetation. And also the fact that, these large buttresses of old growth cypress might impede movement of the small females as they're trying to climb over these. And by small, I mean small relative to river cooters. And so this could, potentially, it seems that abundance is largely influenced to the extent that it's influenced by nesting availability of good nesting locations rather than actual elements of the river, the in-stream river habitat itself. And for Texas river cooter, we see the opposite, right? We see a high level of association with cypress, and we also see a high abundance where the river substrate is mostly bedrock or large and small gravel rather than sandy or silty. So what could cause this? We're thinking to an extent that where you have bedrock and where you have gravel, the river is very clear. It tends to be a lot clearer, less turbid than sandy or silty habitat. So this could increase visibility. Could also increase the amount of sunlight or provide attachment sites for macrophytes, because remember the cooters are herbivorous, so this could influence their foraging, the types of the macrophytes that are available for them to forage on. And it could also just be that better visibility in the water could help them select better plants and also increase and aid in interspecific signaling underwater. We see a lot of interactions with cooters, and perhaps the clearer water where there's more bedrock kind of enhances this. You also see a lot of cooters, which are found in high association with cypress trees. You see them using the flares at the base of cypress trees for basking substrates, whereas you almost never ever see this for Cagle's map turtles. The only time I've ever seen Cagle's map turtles basking on cypress flares are if the cypress is completely submerged in the water. Also, we want to make our models work for us, so we're going to look at latent abundance or density estimates and extrapolations of population size across the different regions of the river. And we were surprised to initially see that habitat was not important for either of these species. All right, so we wanted to look at this further, and we couldn't actually, because of the lack of replication across all levels, we couldn't actually test for a specific interaction effect, but we noticed um, that we saw that Cagle's map turtles in the headwaters region are actually reach the highest densities that they do in the entire system in impoundments. All right, so you might remember um, this is in contrast to previous studies that have determined that map turtles do not like reservoirs, are not found in them, but we see them in high abundance in reservoirs sites, which are primarily what we see in the headwaters. So we see them reaching high abundances. And another thing that's important, though, is that in those major reservoirs in the middle Guadalupe and the lower Guadalupe, like Canyon Lake and the major reservoirs by Seguin, they are actually still present. They're just there in much smaller numbers. All right, so we do see them present. We do see them in all reservoirs where there have been previously presumed to be extirpated. All right, and then for Texana, for the river cooter, we notice peaks of abundance either in headwater spring areas or highly productive lakes. We see that in, for example, the Blanco River here. You see high abundance in the springs at the headwaters. In the lower Guadalupe, you see this kind of transect that has high estimated or predicted abundance. That is actually Kamal Springs, and where we see the highest abundance altogether for cooters happens to be just downstream of the San Marcos Spring Complex. So we see cooters basically occupying um, these spring-fed systems, and this is where they attain high abundances or in headwaters. So this kind of leads us to some estimates of global population size for Cagle's map turtles. So if you look on the right, this is basically a map where the circle size is showing relative densities of turtles. So you can see there's extremely high densities in the headwaters. You could see another cluster of high density location by Gonzales, kind of in the middle Guadalupe or lower. It's technically in our block called the lower Guadalupe. And you also see another kind of extensive area of riverine where it's high abundance down by basically between Cuero and Victoria, right? So you see these kind of different clusters of high abundance, and we have very low abundance, as it turns out, in the middle Guadalupe and in the Blanco River. But collectively, our estimates extrapolated across these geography blocks with the average density for that from our model best fit models for these particular blocks, we get an estimate of about 12,800 map turtles for the Guadalupe San Marcos drainage, which is remarkably similar to the estimate that Killebrew came up with about 25 years ago. All right, so we do have a 50% credible interval of about 8,000 to 21,000 individuals. So you can see that there's a high bit of variation around the abundances. And this makes sense because, of course, we modeled using a binomial distribution, which has high credible intervals. And if you look at how the population is essentially spread across the river, you could see that you could see kind of clusters of high abundance and low abundance. For example, 22% of the population is restricted to the headwaters, whereas you have 65% in the extreme, or I should say in the lower Guadalupe. And you have very low abundance, for example, in the Blanco River, where 2% of the global population is found, and also in the middle Guadalupe, where we only have about 3% of the global population. So if we look at the same thing for Texas river cooter, we have a total study area population estimate, and not global because the species is actually not confined to the Guadalupe-San Marcos River, but we have a total study area population of about 47,650 turtles. You can see there's also high variance in the incredible intervals here, but generally probably about outnumbers Cagle's map turtle by about 3 to 1, which is also very consistent with what Killebrew found. So if you look at the map of abundances on Texas river cooters, you can see high levels, high concentration of abundance at the springs, Camal Springs and San Marcos Springs at the base of the Balcones Escarpment. You can see high abundance basically in all of these kind of Edwards Plateau regions, and kind of a graded decline in abundance as we go down onto the coastal plains. And if we look at this in terms of the proportion of population within the different stretches, you have about 19%, 20, 30, 21. All these percentages are fairly consistent across the different geography blocks. Except that you can obviously see that with the relative, if you look at the relative length of the lower Guadalupe having only 21% of the total population of Texas river cooters, yeah, they're definitely less dense in that area versus the other river reaches. So I think I'm going to skip discussing our occupancy models because this unfortunately took me a lot more time. I was having a very difficult time trying to condense our study into just an hour, which did not work out very well, obviously. But I would like to go to the very end here and talk about kind of our summaries. Because I did not really go through occupancy modeling, I'm just going to tell you right now, you can trust me, I can send you our report, but abundance and occupancy models basically provided complementary insights into both the distribution and abundance of map turtles. We know that habitat features explain some heterogeneity in abundance. For example, map turtles attained higher abundances where terrestrial soils were loam or sandy, and the dominant vegetation was hardwoods. We also noticed a context-dependent influence of impoundments. So impoundments can actually be highly supportive of Graptemys caglei population, and that flowing water is not a requirement. I'd like to emphasize here, though, that in general, at least from my perspective, I would say that I don't want to kind of falsely give the impression that these are habitat specialists. I do think that that would be misleading. These effects from our models are actually very subtle. And I would say that in most cases, if there's enough water, some threshold availability of food, a nice deadwood snag to bask on, and a decent place to lay eggs, you're going to find map turtles will likely be there. Based on the distribution extent that we've looked at for map turtles and our modeling results, we conclude that there's probably stasis in population size over the last 25 years. In other words, they have not been declining. And if anything, although this is once again a conservative perspective, they might actually be increasing in abundances in some areas. For example, in the headwaters region, where we see a lot of low head impoundments that have seemed to support large densities, high densities of Cagle's map turtles. We've also documented a few range extensions into some tributaries in the headwaters region. And what really kind of impressed me about the study is, despite the extreme differences in our methods, and this could of course be complete coincidence, our estimates for actual abundance of Cagle's map turtle and Texas river cooters in the Guadalupe-San Marcos drainage are remarkably similar to what Killebrew found, both in absolute abundance and in relative abundances. And this is despite a huge difference in our detection probability. Our detection probability ranged from about 15 to 30%, whereas Killebrew used a detection correction factor of only 3%. So despite these kind of major differences, we've seemed to come out with very similar numbers. And in particular, both of this current study as well as the Killebrew study determined that river cooters probably outnumber map turtles in the system collectively by about 3 to 1. So what's the best explanation for this? Having such similar estimates despite completely different methodological approaches. And our belief on this is that is in essence that the availability of increased optics have also increased the genuine detection rate of these turtles, right? So we have good binoculars and good cameras now that were not very available 25 years ago, and that seems to be the most parsimonious explanation for why we have similar results despite these huge differences in detection probability. And ours, just for kind of a gut instinct appeal, our detection probabilities range from on Cagle's map turtle from about 15 to 30% basically, which means we had about a 15 to 30% probability of observing the same individual. And we don't have really good ways other than looking at model fit to kind of determine the accuracy of our models, but this feels good based on intuition. We do very clearly see the same turtle on different surveys. For example, you can see this big female with the scar on her head. These were actually from different survey days, although it looks almost identical, right? And I know you might not believe me, but you can tell because if you look at the female, or look at her picture on the left, you could see that she has her shell is still wet where she was dry on the right. So we caught her at a different time in her basking. But also, for example, here in the San Marcos River, this female had very distinctive kind of swirly markings, so she was easy to recognize. So we did see her on multiple occasions, and so basically just being able to identify turtles based on their markings, that 25% detection probability rate feels good intuitively. But of course, this also kind of creates a bias in that there's an anticipatory effect because when we pass the same log, we're obviously going to look for the same turtle, and it's kind of impossible to remove that kind of bias. Just kind of to wrap up here, once again, we see an interesting pattern in reservoirs where we see that Cagle's map turtles are present, but in very low abundance in a lot of the major impoundments of the Guadalupe system. For example, all of these areas, all of these lakes that we've surveyed, Canyon Lake, Lake Placid, Lake Dunlap, Meadow Lake, Starkey Lake, all of them have Cagle's map turtles. All of these are pictures of map turtles from those various lakes. So why are they there but in low abundance? Well, we think too that this could be a bias. They might be in higher abundances than we estimated from our modeling because it's really difficult, surprisingly, to actually survey reservoirs for a lot of reasons. But one of which is, there's a lot of human activity, recreational activity in reservoirs, so you're dealing with a lot of wake overwash and just the effect that people have on turtles. So it's really uncertain whether it's low density, but I have a feeling that if you can see them in the few surveys that we've done in these rivers, or excuse me, in these reservoirs, then then at least we know that they're still there or have come back to those reservoirs. And also the high abundance in the headwaters region, where we see the actual highest abundances of Cagle's map turtles across their entire distribution. What's causing this? I mean, it could be that these types of reservoirs here tended to be more oligotrophic, were likely less polluted. They had lower effluents and cumulative loads because they're here by the spring headwaters. And they did have more productivity in terms of macroinvertebrates from Lexi's data. And they're also very relatively shallow with expansive littoral zones, right? So they have abundant snags. So overall, if you have all of these conditions, it doesn't really matter if the water is flowing or not. You're going to have, you can support potentially high abundances of these map turtles. And yeah, and in terms of the limits of the distribution, if you guys can bear with me for just a couple more minutes here, and then I'll let you go. These red lines show the limits of where we found Cagle's map turtles in the main river system. So we could see them in the upper north and south fork of the Guadalupe, basically to the point of limiting water. Same with the Blanco River upstream of this red line. The river is very ephemeral and intermittent, very little water availability. And downstream, we've observed Cagle's map turtles to about the point of the south side of Victoria. This little black box here, this represents the only area within the distribution where we have not encountered Cagle's map turtles. There was about 4 or 5 surveys there, but this is also the section of river that tended to have the lowest discharge and the lowest flow. So so it was a very difficult area to search, and historically there have been very few sightings from this area as well. In fact, I only know of one sighting site record from this area within the black box, but on either side of that black box, we do see densities pick up. So whether this is real, I think it is real to an extent, although I do, there are definitely turtles there when there are decent conditions. We did find some range extensions. For example, we did find a Cagle's map turtle up about 5 kilometers into Turtle Creek, and very likely they occur much higher upstream than that, but it's a lot of private property, game ranches, and difficult to access. So as far up as we could find access, we did find Cagle's map turtles. We also found them in Johnson Creek, up 18 kilometers upstream from the fork with the Guadalupe. So Johnson Creek was pretty amazing because it's just a very clear water stream, a lot of fun nick points, waterfalls, very narrow places, and a large quantity of map turtles, and a lot of dinosaur tracks. So what more can you want? So anyway, as I kind of wrap this up, what I'd like to say is that we need to kind of potentially revise what we consider to be threats for map turtles for Cagle's map turtle from previous assessments. In particular, I think that the biggest threat to map turtles is continuing climate change and increasing water demands. We have, for example, this is the Guadalupe riverbed at Spring Branch over 2023 and 2024. Look pretty much like this, absolutely no water, right? So this drought that we're experiencing right now basically is the second most severe drought in the last 100 years from this gauge data that we pulled down from Spring Branch. And you can see that even though the duration of this, you can see these red lines kind of indicate periods of extreme drought over the last 100 years. The drought in the 1950s was famous because it lasted about 8 years. However, it didn't get to this low level of discharge until the last year of that drought. And in contrast, in the current drought that we're experiencing now, we've reached levels that were basically comparable to the lowest discharge levels in the 1950s drought, just over 2 or 3 years. So this kind of speaks to the intensity of the droughts now versus potentially over the last century. And in particular, if you combine drought with extreme temperature, this is going to cause and also increase water demands, specifically for related to growth of San Antonio, right? So this is going to create large stretches of the middle Guadalupe River and Blanco River that will be essentially uninhabitable, and in that case they will become extirpated from those stretches, or at least periodically, right? And then the last thing I want to discuss, and I promise I'll let everybody go, are that we also noted in the study that there are Graptemys versa, which is the Texas map turtle, has become established in the upper Guadalupe basin. So we see juveniles, we see adults, we see them consistently, pretty much geographic contiguity over several transects in the headwaters region. So they're not as abundant as Cagle's map turtles, but they're present and they're found. This map on the left shows locations of where we've found and documented Texas map turtles in the Guadalupe. So you can see they kind of extend for a distance of about 40 river kilometers. And importantly, we've also seen individuals that look intermediate. Like, for example, you can see the individuals on the right here have a kind of post-orbital broad kind of bar and these kind of weird kind of concentric oval circles that kind of look exactly intermediate between what you would expect of a Texas map turtle and a Cagle's map turtle. And they often have these little spots on the chin, all right, which is typical of a Texas map turtle. So it's very possible that these two species are hybridizing, and it seems fairly extensively if that's the case. And so this could lead to genomic swamping and kind of eradication of any sort of native genetic diversity in the actual Cagle's map turtle. So we're currently kind of looking at that. Here's a picture of what I think is a hybrid from the North Fork of the Guadalupe. And yes, I know I took a lot of time and I apologize for that. So if you have any questions, if we have time, I don't know, Paul, let me know what you think.
Pardon me. Yeah, we probably should go ahead and wrap up, given
Okay.
we're 20 minutes or so over time. Do you have any contact information you could share? I wonder if people do have questions, they
Yeah,
can follow
absolutely.
up with you. If you have a
Yeah.
slide or anything with that on it, or you could just
Well, I'm easy to contact at Meek, MEIK, at tarleton.edu. So yeah, please, if you have any
people can
questions.
Google you, can't they, and find you on your lab website, I'm sure.
Yes,
Um,
yes. Yeah, absolutely. Yeah.
well, I almost feel bad making you try to attempt to talk about all that information within the confines of one
I feel
webinar.
like, in retrospect, I feel like I should just pick one piece rather than
Well,
try to briefly summarize most all of it. So, yeah,
it was an incredible amount of information, Jesse. So thank you so much for joining us today. I'm going to go ahead and share my screen and just kind of wrap up here real quick.
absolutely.